Charles.
Hello, Shannon. How's it going?
How are you?
I'm good. I'm good. You're safe and healthy and everything?
Yep, safe and healthy. A little bored, but I can handle that.
Today on STEM Thing to Talk About, I've got a good friend of mine, Shannon Murphy, who's going to be talking about her experience as a microbiologist working on her PhD at Cornell University.
So, Shannon, could you start off by just introducing yourself and explaining what it is that you do?
Yeah, so my name is Shannon Murphy, and I am a microbiology researcher at Cornell University. And so what I do is I study bacteria. I study how they normally grow and live, and I try to just learn new things about them. So that when we're trying to devise a new treatment for killing a harmful bacteria, we will have some information to start out on.
Awesome. Now, obviously, so your title is a microbiologist, is that correct?
Yes.
And so when it comes to microorganisms, there's lots of different types of microorganisms, such as archaea and fungi, protozoa, different algae and viruses. What is it that brought you to wanting to study bacteria?
Oh, that's a really good question. So honestly, it was just a lot of the kind of internships and experiences I had leading up during college and afterwards. I just worked with a lot of other scientists that studied bacteria. I just thought that they were really interesting in the different behaviors that they do. So bacteria, they can swim, they can glide, have all different sorts of food sources they can consume. So I was just really excited about the diversity of bacteria that existed.
Huh, that's very cool. The diversity of bacteria and I like how they slide. It sounds like they're dancing around, very active like yourself. So you had mentioned a little bit about some of your prior experiences and like what brought you to like becoming a microbiologist. Um, like, could you explain a little bit more about that? Like, where did you go to college and what did you major in college and what were some of your internship experiences like?
Yeah, so I mean, in high school, I took the normal chemistry, biology courses, and I really liked those. So I knew going into college that I wanted to do something in that area, but I didn't at the time have a clear idea of what I wanted my career to be or what I wanted to do for a living. So I went to a state school in New York called Geneseo, near where you went, and there I majored in biochemistry. So I figured biology, chemistry, I'll put them together. That'll be a winning combo. A lot of my classmates in my courses were interested in going to medical school, but I wanted to do something kind of different. So during the summers in college, I interned in different research laboratories, and I did all sorts of things. One summer I worked with plants, another summer I worked with algae. But what those experiences really gave me was just a taste of what doing research is like. So you'll go in, you usually have like a question or a problem that you're trying to answer, and you just design experiments to figure out how to do that. And I found that kind of problem solving to be really exciting. And from there, I just had to really decide, what kind of organisms do I want to work with? Plants grew really slowly, so I didn't have the patience for those. But microbes grow really, really fast. And so I just loved the rapid pace that research could happen at.
Huh, that's cool. I never thought of it that way. It's true. Like, I mean, that's one of like it's a really cool experiment to watch a plant grow and see how it develops. But I mean, it also can take a really long time. And you had mentioned like how these internships started to bring on different experiences for you and really opened your eyes to the world of research. Now, I think that that's something that can be really cool, but also the world of research from my perception, can be kind of like isolated and not that cool, or not that it's not cool, but just not as exciting, as like being able to interact and stuff. So that just makes me think like, what are some of the things that you like the most and that you like don't necessarily like about like being a microbiologist in a lab?
Yeah, so I'll start with, I think, the things that are the hardest about research. It is a very slow, methodical process, even if you're working with a fast-growing microbe. And there's a lot of failure that you have to contend with. When you start a new experiment in the lab, it will never go perfectly the first time. So you are always trying to refine and optimize the things that you're doing. So you have to be kind of comfortable with trying something out and it not working and being able to bounce back from that and make it better.
Yeah, being comfortable with failure, absolutely.
That's really just one of the hardest parts. But I think with that, the excitement of doing research and the thing that has driven me towards a research career is that feeling of excitement when something does work out. When you have an idea, you test it, it turns out to be either what you thought or something completely different that's new. That's just such an exciting feeling of like you've discovered something that nobody else knew about. And it's that feeling that you're always chasing, and that's what helps you get you through kind of the days and weeks and months and sometimes years of things not panning out.
Yeah, absolutely. And I mean, throughout all that, like the failures and the successes as well, how do you keep track of like all of this, like this plethora of experiments and information that you're taking in? I feel like, the world of biology and biochemistry, like, and then you narrow it down a little bit more to what you're doing, even what in that narrow field that you're in, or like narrow field that you're in, like there's still so much, like I feel like that's got to be so overwhelming. Like what are some of the techniques and whatnot that you do to like organize your experiments and really like organize your thoughts of what it is that you want to experiment with?
Yeah, so organization in science in general is really important. For a number of reasons. Like one, when you do an experiment, you have to write enough detail about it that somebody else could pick up your work and they could do the exact same thing. Or sometimes more commonly, you might just have to pick up and do the same thing a week later, and it's amazing how much you forget in a week if you don't write it down. So just being able to have reproducible science, that's like one way that you can convince other scientists that it's real. Can they get the same outcome in their lab with their hands? So what all research has is we have a notebook. Like it's literally just a journal that we write every detail down of what we do. And it's often kind of messy, you might have multiple experiments going on at the same time, but that's our raw sort of log of what's happening. So from there, what we do to organize is we'll usually type up that information. We'll have databases where we can, share it with the other people we work with. And ultimately the goal of that is to, write up kind of a complete story or project that we can then share and send out to other scientists.
And within that world of research, something that I had watched one of your videos before we started talking here, and one of the things that I thought that you mentioned was so cool is like in the world of basic research, so much of what you do is question, and I think that is so empowering and awesome that in your lab, that's what you do is you question things and you start to research it. Could you explain like what are some of the projects and experiments that you started working with? I know that your lab does a lot of different work with bacteria, but what is it that you specifically have taken looks at in the past?
So one of the things that I study is actually just a specific structure on the bacteria. So bacteria, in order to, kind of keep themselves contained from the outside world, is they have this cell wall. And this wall is really important because if there's any damage to it, the bacteria will just sort of pop like a balloon. And one cool thing about this wall is that because it is vulnerable, the bacteria are vulnerable without it, we have a lot of antibiotics that we can use that will disrupt the wall and then the bacteria will die. And that's how we treat people who have bacterial infections. But one problem that we're encountering is that bacteria are gaining resistance. So they have different adaptations that will protect them from the antibiotic. It'll help them keep their wall stable. So what I'm just studying is at a basic level is how do bacteria build this wall? How do they defend it against different drugs? And how do they just remodel it as they sort of grow and divide and do their thing?
Yes. And so with these antibiotics, like what is happening with these antibiotics that you really have to like that really is the big concern within like our society and the medical world?
Yeah, so, we've been using antibiotics for almost 100 years, and they've worked really well. Like they revolutionized modern medicine because an antibiotic can specifically kill a bacteria, but it doesn't harm our cells. But the problem is that bacteria are able to evolve very rapidly. So they can just have one genetic change that can go from being susceptible to this drug to now they're resistant. And that becomes a problem when we have infections that resist 1, 2, dozens of different antibiotics that we have. So that's a problem that microbiology researchers are trying to address, is figuring out how can we better target this bacteria so that our antibiotics work better, or how can we find a new target to design a new antibiotic.
Mm-hmm. Yeah. And I mean, like that's the term that they use is like a superbug, right? Like for a bacteria that is gonna that's more antibiotic resistant. So definitely something that's scary. What are some ways that that like a common person who's not a microbiologist can work to not become or to not have bacteria within them that is resistant to antibiotics?
That's a really good question. So the biggest thing is to prevent yourself from getting sick in the first place. So that just goes back to basic hygiene of good hand washing, especially if you've been in public places. And that's really, I mean, topical today when we're talking about not just bacterial infections, but also viral infections. But inevitably, we do get sick. One of the most important things is, if you visit a doctor, they will prescribe you an antibiotic only if you have a bacterial infection. What a lot of people don't understand is that these antibiotics, if you have a viral infection, which sometimes the symptoms are very similar, the antibiotic won't work. So one kind of big problem is if you are using antibiotics when you shouldn't be, that can help kind of grow antibiotic resistance within you. So the next time you took that same drug, it might not work as well. But if you are in a position and your doctor prescribes you this and you need it, you should take it, and you should take the full course of it, exactly as they've told you, and that can help prevent antibiotic resistance from kind of growing and developing.
Mm-hmm. Absolutely. And I think that's something that people will be like, oh, like I feel better now, I can stop taking it, but it doesn't necessarily completely get rid of that bacteria. Then that gives it a chance to evolve, like you mentioned, because it can happen so quickly.
Exactly.
Now, as we mentioned before, a microbiologist can study lots of different types of microorganisms. One of those being a virus, which right now is pretty current events and pretty applicable as we're all wondering and reading the news about the coronavirus. But you do not work with that. Is that correct?
Mm-hmm.
Yeah.
And there's plenty of other research labs at Cornell that are working on coronavirus, both in trying to develop new treatments as well as vaccines. So they're very hard at work right now.
Mm-hmm. Yeah, but I think that's also something that's super important to recognize is that you can't give an antibiotic is not going to treat the coronavirus, and I think or any viral infection. So just because you're sick does not mean an antibiotic is something that you need, because a lot of times bacteria can actually be good for you too, right? So like we've got probiotics, which obviously your body needs it also in order to stay healthy. Like antibiotics are not always going to be the cure, which I think is important for.
And you made another point there too, like not all bacteria are bad. Bacteria are in our environment everywhere, and only a fraction of them actually make us sick. I think one of my favorite examples of microbes that are helpful are in food. So a lot of the dairy products that you eat are produced using microbes. Yogurt, cheeses. So there's a lot of good things that they can do for us too. But I tend to only study the harmful ones.
Yeah, those are the really fun ones. What is the most harmful bacteria that you have worked with?
So I've worked with a number of different ones. The main bacteria that I work with that is pretty dangerous is the one that causes cholera. So this is a diarrheal pathogen, and it can be potentially lethal just because it can dehydrate you very quickly. And it is a problem mostly in developing countries where people don't have access to safe drinking water. Because if they drink water that's contaminated, it will start an infection and it will spread that way. So a few million people develop cholera every year, and a small fraction of those do result in death. So it is a pretty dangerous pathogen. That being said, there's a lot of precautions that we take in the lab to work with it to make sure it's safe. So there are governing bodies that sort of divide up bacteria into different categories to kind of rate them on how dangerous they are. And some of the factors that go into play there are how easy is it to spread from person to person? How sick would you become if you did contract an infection? Things like that. So there's a whole scale of that, and that will dictate the precautions you take. So in the lab, I wear gloves. There's no food or drink in the lab. We sometimes wear lab coats. We sometimes work in hoods, things like that, just to make sure that there's a good barrier and protection between us and the organism we're working with.
Mm-hmm. Besides personal protective equipment, what is some of the equipment and tools that you do use in your research?
Oh, to study bacteria?
Yeah, to actually study the bacteria. And like, could you explain a little bit about those experiments? How, like, what does that process kind of look like?
Yeah. So, I mean, one of the main tools we use as microbiologists are microscopes. So some of them are very simple. They're just using light in order to magnify the bacteria so we can see them. But there are even more advanced microscopes that don't rely on light, that rely on, electrons and other things where you can get really detailed images of the bacteria. So we do just try to look at them. We can see, their shape. We can see how they move. But there's also a lot of other tools that we use. So one of our main kind of techniques for figuring out how a bacteria is doing something is we will actually take a look at their DNA. So we will sequence their DNA. This is just the code that, gives bacteria and us, just the traits that make us who we are. And so we look at the bacterial code. We can actually make changes to it so through some pretty high-tech techniques. And usually what we're doing is we're knocking out, we're just deleting a specific sequence in there and just seeing what happens to the bacteria. Sometimes it might stop them from swimming properly, and then we know that that region of code helps them to swim. It might mess up the shape of them, and then we know it's important for keeping them into a sphere or into a rod shape. So we really rely on creating mutant versions of the bacteria to then kind of work backwards and think, like, what is that DNA normally good for?
Mm-hmm. Because that DNA is essentially the directions for the microbacteria functioning. And like, there's so much for it. A bacteria is a type of prokaryotic cell. Is that correct?
Yes.
Awesome. And so how does that differ from a human cell?
Ooh, so there's a lot of structural differences between prokaryotic and eukaryotic cells. So a lot of prokaryotes, because they don't have, arms and legs and organ systems, they have to kind of arrange things differently. So they might have a small tail on the cell that will help them to move. They also kind of, things are more loose, they're less organized than like humans that might have organ systems. And also just to maintain some structure, bacteria will have, in addition to like a squishy membrane that holds everything in, they will also have a cell wall, and that will give them like a defined shape and a structure that, other eukaryotic or multicellular organisms can impose in other ways.
Mm-hmm. And I mean, that cell wall of the bacteria is really like, that's a major part of your research. Am I correct? That cell wall, and like, I thought that you put it excellently in your video, how like the bacteria is the enemy and you guys are studying the enemy, and it's like that cell wall is like the bacteria's protective mechanism. And so if you guys are able to figure out more about that through your study in the lab, then we could potentially find ways to prevent it from becoming resilient to antibiotics.
Exactly. Yeah, and I mean, that cell wall, it's interesting because it's, in one ways, it's bacteria's best defense, but it's also their greatest weakness. And the reason that, antibiotics work is because they target a cell wall, and our cells don't have them, but bacteria cells do. So it's that difference that really makes them both strong, but also vulnerable. So we're really looking for breaks in that kind of armor and seeing how can we better destroy it.
Breaking the armor, destroying it. Beautiful thing. It's awesome. If you had advice for a middle school student who is interested in something like microbiology or biochemistry, what would be your advice for them?
Yeah, I would say work hard in all of your classes, because you'll never know, what kind of background information your career might need. So for my day-to-day job in microbiology, it uses a little bit of everything. So I rely on a lot of chemistry, because we're looking at, a lot of drugs and interactions and how we can chemically manipulate microbes. We're also looking at a lot of genetics is the main part of that, and you'll get that in some of your biology courses. Another big part about it that I didn't expect to need to know was math and coding. So you're creating a lot of data as a scientist, and sometimes it's too much for one person to go through it all by hand. So what you do is you train a computer to analyze that for you. So that's a skill that wasn't on my radar that I had to kind of learn later in the game. So I would say just be open to sort of explore, learning all that you can, because you never know when it might come in use. And mostly, especially in research careers, if you get to a point where you haven't had that experience, that's when you learn. So you just have to be open to learning new things and kind of embracing them when they come up.
Thanks, Shannon, for coming on the podcast today. We really appreciate you.
Yeah, thanks for having me.
This has been another episode of STEM Thing to Talk About. I'm your host, Charles Rumlard. Tune in next week for another episode.